Hello everyone. Welcome to the session of Sexual Reproduction in Flowering Plants NCERT exercise part 1. So today we are going to solve the NCERT exercise from the chapter Sexual Reproduction in Flowering Plants, and today is the first part of this NCERT exercise. Disclaimer: the images, text, and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issue, please contact us at sathee@iitk.ac.in and we shall remove the content immediately. Please note that the parts of the content may have been generated using the generative AI, which is totally experimental. So, what is the learning objective? To practice and master the core concept covered in the NCERT exercise through problem solving. So, we are going to solve each and every questions from the NCERT exercise. And today's questions are from the section 1.2 from the chapter Sexual Reproduction in Flowering Plants. So, here is the first question. Name the parts of angiosperm flower in which development of male and female gametophyte takes place, right? So you have to tell the male and female gametophyte, where at which part of the flower this male and female gametophyte takes place. The question comes from the section 1.2, means the question is based on the section 1.2. So this is the question, right? Name the parts of an angiosperm flower in which the development of the male and female gametophyte takes place, right? So let us see the answer of it. The male gametophyte or the pollen grains. Pollen grains are what? They are the male gametophyte, right? Where does it develops? It develops inside the pollen chamber of the anther. It develops inside the pollen chamber of the anther. Whereas the female gametophyte, and what we call the female gametophyte? Embryo sac. Embryo sac. So that develops inside the nucellus of the ovule. Nucellus of the ovule from the functional megaspore. So this functional megaspore, one of the functional megaspore, right? That forms the embryo sac. And this functional megaspore actually from where? One of the nucellus cell undergo reduction division or the meiosis division, and it forms the 4 megaspore. Out of these 4, the 3 get degenerated and only one becomes the functional. So that functional megaspore give rise to this embryo sac structure, right? We have seen in our theory classes that how does it forms, right? So here you have to name the parts of the angiosperm flower in which the development of the male and female gametophytes takes place. So this is the answer of that question. Let's move to our next question. Differentiate between the microsporogenesis and megasporogenesis. You have to differentiate this. And it has another question also. Which type of cell division occur during these events? Like they are asking which type of cell division that occur in the microsporogenesis and megasporogenesis, right? Name the structures formed at the end of these two events, and you have to also name the what is formed at the end of these two events, that is the microsporogenesis and megasporogenesis. Okay, so this question also comes, or you can say this question is based on the NCERT section 1.2. So first of all, you have to differentiate microsporogenesis and megasporogenesis, right? So let us see the answer of this question. So this is a question: differentiate between microsporogenesis and megasporogenesis. So let us see the first answer, because there are three different questions. So the first answer, microsporogenesis is the process of the formation of microspore tetrad from the microspore mother cell through meiosis, right? Okay. So what is formed and from which cell? So here, the first cell that you see is the microspore mother cell. From where this microspore mother cell get develops? From which tissue? From which tissue this microspore mother cell develop? This we must know. Which of the tissue that forms the microspore mother cell? Inside the microsporangium, or you can say the theca. First of all, we have studied about the theca, and what tissue is present inside the theca? Sporogenous tissue. Sporogenous tissue, right? Now, all the cells of the sporogenous tissue is having the capacity to form the microspore mother cell. So those tissue, the cells of those tissue become differentiated as they accumulate a lot of cytoplasm. The nucleus become very prominent, right? And that cell is ready to undergo the meiosis division. And that cell is called as microspore mother cell, right? So this microspore mother cell undergo meiosis division, and what they form? They form microspore tetrad. They forms the microspore tetrad. What is the process? What is the process that calls the formation of this microspore tetrad? Through the meiosis division. So the microspore mother cell, MMC, undergo meiosis division and it forms the microspore tetrad. This whole process is called as microsporogenesis. This whole process is called as microsporogenesis. Now we will see how it occur. What is the next point for this megasporogenesis and how it is different from the microsporogenesis. So the megasporogenesis is the process of the formation of 4 megaspores from megaspore mother cell in the region of the nucleus through the meiosis. So it is same thing, almost the same thing, but still there is a difference. Here there are number of microspore mother cell, but here the megaspore mother cell is just one megaspore mother cell. Normally they are having only one megaspore mother cell, but microspore mother cells are many in number. All the sporogenous tissue is having the capacity to form this microspore mother cell, right? But the cells of the nucleus, one of the cells of the nucleus transform itself and undergo the megaspore mother cell. It undergoes megaspore mother cell. This megaspore mother cell is still diploid. It undergo meiosis division and it forms the 4 megaspores. Now this whole process, that is the formation of megaspores from the megaspore mother cell through the meiosis division is called as megasporogenesis. So this is very, very important difference. Microsporogenesis occur inside the pollen sac of the anther. This is the position. It occur inside the pollen sac of the anther, whereas the megasporogenesis occur inside the ovule. Right? This is also one of the difference, that microsporogenesis occur inside the pollen sac, whereas the megasporogenesis occur inside the ovule. And where in the ovule? You must know. Where in the ovule? Inside the nucellus. One of the nucellus cell get transformed into the megaspore mother cell, and that undergo reduction division or the meiosis division and form the 4 megaspores. Now, both the events Both the events like microsporogenesis and megasporogenesis involve the process of which division? Because the second question, read the second question. Which type of cell division occur during these events? So, it involves the process of meiosis or reduction division, which results in the formation of haploid gametes. Okay? It results in the formation of haploid gametes from the microspore and megaspore mother cell, right? Microsporogenesis result in the formation of haploid microspores from the diploid microspore mother cell. This microspore mother cell that we call it MMC, microspore mother cell, right? That undergo reduction division or the meiosis, and it forms the haploid microspores. Now, on the other hand, if you see in the megasporogenesis, the result is the formation of haploid megaspores from the diploid megaspore mother cells. Diploid megaspore mother cell. So, this was the question. Name the structure formed at the end of these two events. What are these two events? Microsporogenesis and megasporogenesis. So, the end product of the microsporogenesis is the haploid microspores, and the end product of the megasporogenesis is the haploid megaspores. This you should remember. Okay, so this is the elaborated answer for all these three questions. That is the question number 2 from the section 1.2. Okay, let us move to our next question. The next question is very important. Arrange the following terms in the correct developmental sequence. So, if you are clear about the microsporogenesis or the megasporogenesis, then you can make a sequence of the cells that are formed in the process, right? So, here are some of the developmental stages are given. The developmental stages are given for the microsporogenesis, right? Pollen grain, sporogenous tissue, microspore tetrad, pollen mother cell, and the male gamete, right? All the stages are given. You have to arrange in a proper developmental sequence. And this is the NCERT question from the section 1.2, right? Now you can think about such question also comes in your NEET examination, that you have to, the 4 options were there in the sequence. All the possible sequences are given, but only one is correct, right? So such questions also comes in the NEET examination. You can think about for the megasporogenesis also. Here the sequence, you have to make the sequence for the microsporogenesis process, but that could be asked also for the megasporogenesis. Okay, without wasting time, let us see what is the correct answer of this question. So this was the question. Arrange the following terms in the correct developmental sequence. So which part you will keep the first one? Which part you will keep the first one? Okay, let us see the correct answer. The correct development sequence is as follows. Sporogenous tissue, and one of the cells of the sporogenous tissue, they get transformed into the pollen mother cell, that is PMC, pollen mother cell, right? And then this pollen mother cell PMC undergoes reduction division or the meiosis process, and what is formed? Tetrad structure, microspore tetrad structure is formed, right? Microspore tetrad structure is formed. And all these tetrad structures are what? The 4 pollen grains, but they are joined together because their walls are not separated. So, after this microspore tetrad, pollen grain is formed. And you all know that the pollen grain is having the 2 cells, vegetative cells and generative cells. The generative sorry, the generative cells divide and that give rise to the two male gametes. So the male gamete comes at the last stage, right? So during the development of the microsporangium, each cell of the sporogenous tissue act as a pollen mother cell. Very important line. This line is very important. Each cell of the sporogenous tissue act as a pollen mother cell, right? But what about the nucellus? In the case of nucellus, only one cell become differentiated as a diploid cell, that is the megaspore mother cell, that is the megaspore mother cell. But here you can see the sporogenous tissue, all the sporogenous tissue act as a pollen mother cell, right? Okay, and that give rise to the microspore tetrad. This pollen mother cell undergo the meiosis division and that give rise to the microspore tetrad, that containing the 4 haploid microspore by the process of meiosis, microsporogenesis, right? Now, as the anther mature, these microspores are dissociate and develops into the pollen grain, right? The microspore tetrad is formed at the meiosis process from the your diploid microspore mother cell, right? And when these microspores are dissociated, they are separated. The tetrad structure, all the pollen grains get separated and they will form the pollen grains. So the pollen grain mature and they give rise to the male gamete, right? So this pollen grain, mature pollen grain, they are having the generative cells and the vegetative cell. When they germinate on the stigma, they give a pollen tube like a structure, and inside the pollen tube, the generative cell nucleus divide into two male gamete, right? Next question is here. With a neat label diagram, describe the parts of a typical angiosperm ovule. Okay, so this is a like total question, that is you have to make a diagram, right? So diagram of ovule. So this question is again from 1.2 section. So an ovule is a female megasporangium where the formation of megaspore takes place, right? You all know that the female megasporangium is the ovule. Ovule is also called as female megasporangium, and where these megaspores takes place in the ovule only. Inside the ovule, the megaspores are formed. So here you can see the ovule structure. So the ovule, when you see, there is a stalk-like structure. First of all, there is a stalk-like structure that is called as funicle. And there is an oval body that is the main part of the ovule. Oval body, that is the main part of the ovule. This main part of the ovule is joined with the stalk-like structure that is called as funicle. Okay, this is the funicle, stalk-like structure, and the main body is joined with it. Where it joins, there is a clear-cut demarcation, and that is called as hilum. Now, we will see the main part of the ovule. Okay, you can see there is a two overlapping covering like this, the first and the second. Similarly here also, first and second, the inner and the outer. This is actually the integuments. The inner integument and the outer integument. This inner and outer integument is actually the outgrowth of the nucellus from the chalazal pole. Okay, it is extended from the chalazal pole and there is an outgrowth from this side to this side, right? So this is the outer integument and inner integument. What is present between inside the integument? The nucellus, very important, nucellus cells. Okay, nucellus cells are present. The nucleus cells are diploid. First of all, they are diploid, and one of the nucleus cells get differentiated from the other cell, and later on it forms the embryo sac. And you all know how it forms. That cell undergo the meiosis division, and it formed the 4 megaspore. Out of the 4 megaspore, out of the 4 megaspore, only one is functional. Only one is functional. Three are non-functional, right? Three get degenerated, and that one functional one only called form this embryo sac. This forms the embryo sac. Okay. Now here you can see the micropilar pole and chalazal pole. Micropilar pole and chalazal pole are there, right? At the chalazal pole, you can see this integument start divide and moves towards this nucleus side, right? And at the micropilar pole, how this pole is formed? This pole is formed by the integument. The integument from both the sides, they unite, but at one particular point, they do not get joined, and there is a suture, or you can say a pore like structure is present, that is called as micropyle. So this is the whole structure of the you can say the megasporangium or ovule. So don't get confused with these two words, megasporangium or ovule. That is the same name. So the various parts of the of an ovule are: first one is the funiculus. It is a stalk-like structure which represents the point of attachment of the ovule to the placenta of the ovary. Right? This is a stalk-like structure which represents the point of attachment of the ovule to the placenta of the ovary. Hilum, it is the point where the body of the ovule is attached with the funiculus. So that particular point is called as hilum. We have seen in the diagram also, right? Hilum, where the hilum is present. Integument, they are the outer layer surrounding of the ovule that provide protection to the developing embryo. So all these structure, if I ask about the ploidy, what is the ploidy of funiculus? So funiculus is diploid structure, right? Ploidy of hilum, this is also the diploid. The ploidy of integuments, this is also diploid, right? All the cells are diploid here. Now the micropyle is not, you can say this is just the pore like structure, right? It is a narrow pore formed by the projection of the integuments. From both the side, the nucleus is covered by the integument, the outer and inner integument, and this integument is not joined at one particular point. That is the micropyle, right? So it makes the point where the pollen tube enters the ovule at the time of fertilization. But this pore is very, very important, because through this pore only the pollen tube which emerges out of the pollen grain, right? It get enter inside the ovule through this pore only. And because of this, the fertilization process occur, right? So this is very important. Nucellus, it is a mass of the parenchymatous tissue, very important. It is a mass of the parenchymatous tissue surrounded by the integument from the outside. Integument surrounds the parenchymatous tissue, that is called as the nucellus. The nucellus provide nutrition to the developing embryo. So this is the function of the nucellus. So when the embryo get developed, all these cells provide nourishment to the developing embryo. The embryo sac. Now, what is the ploidy of this embryo sac? Embryo sac is located inside the nucellus. So, first of all, the ploidy of the nucellus cells are diploid. The nucellus cells are diploid. But what is the ploidy of the embryo sac? It is a haploid structure. It is a haploid structure. So remember all these points, these are very, very important. So this ploidy is not asked in this question, but yes, I'm telling you this because it could be asked in some other questions, right? Okay. Chalazal pole, it is a based basal part, swollen part of the nucellus from where the integument originates. So again, you got a very important point. From where the integuments originate? It originate from the chalazal pole. It's originate from the chalazal pole. Okay. So this is for all these points are for this question number 4. Okay. We have discussed about the diagram and the different parts also. Okay, moving to our next question. The next question is, what is meant by the monosporic development of the female gametophyte? One of the very important question from this particular chapter. Monosporic development in your NCERT, it is properly mentioned that such type of development is called as monosporic development for the female gametophyte. So this also comes from the NCERT section 1.2. Let us see the answer of this question. What is meant by the monosporic development of the female gametophyte? The female gametophyte or the embryo sac develops from a single functional megaspore. Single functional megaspore. How many megaspores are formed? 4 megaspores are formed like this. 1, 2, 3 and 4. 4 megaspores were there, right? And out of these three get degenerated and only one is functional. So 1 megaspore is functional, right? 1 megaspore. So this is known as monosporic development because only 1 megaspore involved in the formation of embryo sac or the female gametophyte structure. And how many cells are there in embryo sac? How many cells are there in the embryo sac? In an embryo sac, how many cells and the nucleus are there? There are 7 cells and 8 nucleate structure. This embryo sac, or you can say this female gametophyte, that is the same name, female gametophyte and embryo sac, right? They are the 7 cell and 8 nucleus structure. 7 cell and 8 nucleate structure. And this all 7 cell 8 nucleus structure is formed from one functional megaspore. And that is why it is called as monosporic development of the female gametophyte, because only one megaspore is involved in the formation of 7-celled 8 nucleate structure, that is female gametophyte or the embryo. So here you can see this is the structure of an embryo sac. Okay, an embryo sac, you can see it is having the 3 antipodal cells at the chalazal end. It is having at the micropylar end, they are having the egg apparatus. 1 egg and the 2 synergid, that unitedly called as the egg apparatus. And there is a 1 central cell. This is the central cell. This cell is the central cell. Inside the central cell, there are 2 nucleus, 2 polar nuclei are there. So count the total number of cell and the nucleus. So we are having 7 cells, 3 at the chalazal end, 3 at the micropylar end and 1 central cell. So 7 cells. And count the number of nucleus. 3 nucleus are present in the antipodals, 3 nucleus are present in the egg apparatus and 2 in the central cell. So total 8 nucleus, right? All these structure, this all structure is formed from the one functional megaspore, and that is why it is called as monosporic development. This is called as monosporic development. In most flowering plant, a single megaspore mother cell present in the micropylar pole of the nucellus region of an ovule undergo meiosis to produce 4 haploid megaspores. So they are telling the same thing, that at the micropylar pole, one of the nucellus cell, okay, one of the nucellus cell, that is called as megaspore mother cell, undergo reduction division or meiosis division, and that forms the 4 haploid megaspore, right? So out of these 4 megaspores, only one functional megaspore develop. Only one functional megaspore develop, right? And that develops into the female gametophyte. Just now we have seen that particular diagram, embryo sac diagram, that is also called as a female gametophyte, right? While the remaining three degenerate. Okay, 4 haploid megaspores are there, and out of that 3 get degenerated, right? So this structure is formed from only one functional megaspore. Next question, question number 6. With a neat diagram, explain the 7-celled 8 nucleate nature of the female gametophyte. This question also comes from the section 1.2. Okay, so you have to draw a neat diagram, and that explains the 7-cell 8 nucleate structure. So this is the diagram that you have to draw. While this is the picture representation from the NCERT book, but when you will draw like this only, that you will write up that there are the chalazal end, there is a micropylar end, right? So this is very, very important to make the two end of the embryo sac, the chalazal end and the micropylar end. At the chalazal end, you will draw the 3 antipodal cells, okay, 3 antipodal cells and 3 cells. You can see the synergid cells are quite large cell, and egg is present behind this. This is the egg cell. Okay. Now, a very important point here, the synergid, you can see the finger-like projection inside the synergid cell. That is called as filiform apparatus. And there is one central cell which contains the 2 polar nuclei. Now, what is the ploidy? I'm asking a different question from this particular question. What is the ploidy of each cell? What is the ploidy of antipodal cell? What is the ploidy of central cell? What is the ploidy of egg? What is the ploidy of synergid? All are having n number of chromosome, means haploid. They all are haploid because they have been developed from one functional megaspore, and that functional megaspore was haploid. So when this functional megaspore nucleus divide and it forms the 8 nucleate structure, and later on this 8 nucleate structure transformed into the 7-celled and 8 nucleate structure. So this is that particular female gametophyte or the embryo sac. So all the cells you see here in the case of embryo sac are haploid. So don't get confused. So I am just repeating one more point. Okay. Megaspore mother cell, one of the nucellus cell get differentiated, that was diploid, that undergo meiosis, form 4 megaspore. Out of these 4 megaspore, 3 get degenerated. Only one functional megaspore is there. That involved in the formation of this female gametophyte 7-celled 8 nucleate structure. The female gametophyte embryo sac develops from the single functional megaspore, and that functional megaspore is haploid in nature. This megaspore undergoes the 3 successive mitotic division. Mitotic division is a simple division, right? Mitotic division to form the 8 nucleate embryo sac. The first meiotic division in the megaspore forms the 2 nuclei. When the first meiotic division occur in the megaspore, it forms the 2 nuclei. And one nucleus move toward the micropylar end, while the other nucleus move toward the chalazal end. Then these nuclei divide at their respective ends. They divide and redivide and forms 8 nucleate stage. Total 8 nucleate. 4 at each pole. 4 nucleus at each pole. So as a result, there are 4 nuclei at both the ends, and that is at the micropylar end and the chalazal end in the embryo sac. At the micropylar end, out of the 4 nuclei, only 3 differentiate into 2 synergid and 1 egg cell, right? Together, they are called egg apparatus. So, egg apparatus? Sometimes it can be only asked in one liner question, 1 marks question, very short answer question. What is egg apparatus? So what you will write? Egg apparatus is a part of the embryo sac or the female gametophyte, and that constitute one egg and the two synergid cell, right? Similarly, at the chalaza end, there 3 out of 4 nuclei differentiate as the antipodal cell. So totally there are 4 nucleus at the chalaza end, right? But one nucleus move toward the central cell, okay, toward the central cell, and that forms the polar nuclei. The rest 3 get differentiated as an antipodal cells. The remaining 2 cells, micropylar and chalaza end, move toward the center and are known as the polar nuclei, which is situated in the large central cell, which is situated as a large central cell. Hence, at maturity, the female gametophyte appears as a 7-celled structure though it has 8 nucleate. It has 8 nucleus. Okay, moving to our next question. This is a very important question, and we have discussed when we were discussing about the theory part of the chapter, right? What are chasmogamous flower? Can cross pollination occur in clistogamous flower? Give reason for your answer. Okay, 3 questions are there, right? And this question also comes from the section 1.2. So let us see the answer of this question. What is a chasmogamous flower? There are 2 types of flower present in the plants, namely oxalis and viola. Chasmogamous and clistogamous flower, 2 flowers. So there are 2 names, 2 species names are given, oxalis and viola. Can anyone give me the third name of the plant that bears the 2 different types of flowers, chasmogamous and clistogamous flower? Both the flowers are present in the same plant. So 2 examples are here, oxalis and viola. Can anyone give me the third name? Okay, so I'll give the third name. There are some examples of the plants that give 2 different types of flowers, chasmogamous and clistogamous. So I got the name Comelina. Very good. The third flower is the Comelina. And in the NCERT book, the figure is given that bears the 2 different types of flower in the same plant. And that figure is from the Comelina only. Okay. Anyway, so the question was about the chasmogamous flower. What is chasmogamous flower? Chasmogamous flower have exposed anther and the stigma similar to the flower of other species. So which flower has exposed anther and the stigma? The flower that blooms, the flower that opens, and they exposed their anther and stigma. If the flower is closed, then their stigma and the anther will not be exposed, right? So this point is there. Exposed anther and stigma. This means that flower is blooming, flower is opening, the bud is opening, and there is a clear-cut flower that is having exposed anther and stigma. That flower is having exposed anther and stigma. So that type of flower are called as chasmogamous flower. Chasmogamous flower, right? And these flowers are present at the shoot only, shoot part of the plant, right? Now we are having the another type of flower that is called as clistogamous flower. The flower that do not get open, they are present in the bud form only. They are present in the bud form only. So chasmogamous flower present on the shoot and it get open, expose anthers and stigma. Clistogamous flower present at the root system, right? And they are in the form of bud. They do not get open, so their anthers and stigma are not exposed, right? So cross pollination cannot occur in the clistogamous flower. So this was the second question answer. Can cross pollination occur in the clistogamous flower? No. There cannot be the cross pollination in the clistogamous flower. Why? What is the reason? Because the flower do not get open. The flower is in the closed form only. Okay, it is in the form of bud only. Their anther and stigma are not exposed. So how could this cross pollination occur? So this is because the clistogamous flower never open at all. Also, the anther and stigma lie close to each other in these flower because the flower is closed na, in the form of bud. So their anther and stigma are very near to each other. They are very close to each other. So they help in the process of self-pollination. So in the case of clistogamous flower, self-pollination is flower possible, whereas in the case of chesmogamous flower, cross-pollination is favorable. Next question, question number 8. Mention 2 strategies evolved to prevent the self-pollination in flower. This is very important question. Very important question. You have to find out the 2 strategies that plant itself evolve in their flower to prevent self-pollination. They do not want that self-pollination occur in the flower. So they, in the due course of evolution, they developed such strategies so that the self-pollination should be stopped or could not take place, right? Let us see what are those 2 strategies. So this was the question and let us see the answer. Self-pollination involves the transfer of pollen from the stamen to the pistil of the same flower. Now this point is very important. Same flower. The pollination is occurring from the stamen part to the pistil part. Stamen part to the pistil part means from the anther the pollen grain is released and it falls on the stigma of the same flower. So this is the self-pollination. Now, the 2 strategies that have evolved to prevent this self-pollination in the flower are as follows. So, there are some strategies that flower itself develops so that no self-pollination should occur. I will explain why they want this. Why they want this. First point, in certain plants, the stigma of the flower has the capability to prevent the germination of pollen grains and hence prevent the growth of the pollen tube. Okay? Stigma of the flower, they are having the capacity to prevent the germination of pollen grain. They do not allow the pollen grain to germinate on the stigma and hence prevent the growth of the pollen tube. So if there is no germination, then how the pollen tube could come out, right? So it is a genetic mechanism to prevent the self-pollination and called self-incompatibility. In the last class, I have told you that there are genetic mechanism. What is the genetic mechanism? There are, if the word genetic comes now, then there is an involvement of genes. I told you that in the case, suppose you are taking an example that pistil is having gene like S1 and S2. And the pollen grain is having also the gene, same gene, S1 and S2. Then this pistil will not allow the pollen to germinate on its stigma. Why so? Because they are having the same type of gene. They are having the same type of gene. And the pollen, this stigma do not allow the same type of pollen grain to germinate. If the pollen grain is of S3 or S4 type, then they will allow this pollen grain to germinate, right? So this is the self-incompatibility, right? So it is a genetic mechanism to prevent the self-pollination that is called as self-incompatibility. Incompatibility may be between the individual of the same species or between the individual of the different species. Different species means you are having the rose flower and the pollen grain from the sunflower is coming on the rose flower. So this is incompatible. Incompatible. You are having the rose flower, having the pistil S1 and S2 type, and the pollen grain is coming and falling on the stigma. And if the gene is having the S1 type, then this pistil will not allow the S1 pollen to germinate. Another pollen is of S4 type. So this is the different pollen of the same species. So they will allow this S4 pollen to germinate on it, right? So this is written here exactly. Thus, the incompatibility prevents the breeding, right? Second point, you have to tell the two strategies. So one strategy was self-incompatibility. Now let us see the second strategy. In some plants, the gynoecium mature before the androecium or vice versa. Means the gynoecium mature before the androecium. The gynoecium matures. The gynoecium matures before the androecium. Androecium is not mature at that time. So, if the pollen coming from this androecium, okay, that will be immature type. So, self pollination is discouraged here, right? So, there is two phenomena that is called as protogyny and protoendry. Protogyny and protoendry. So, here in the case of protendry, what happens? In the case of protendry, androecium matures earlier and gynoecium matures later in the same flower. So the maturation time of the male and female reproductive organ are different. So in this way, they prevent the self-pollination. Protogyny. Gynoecium mature earlier and androecium mature later. So again, they are discouraging the self-pollination in this way. So this prevent the pollen from coming in contact with the stigma of the same flower, right? So these are the two different mechanisms in which the flower evolve itself and prevent the self-pollination in flower. Why they don't allow the self-pollination to occur? Because self-pollination will bring inbreeding depression. Inbreeding depression. No new variation will be originated. No new variation you will see in the plants. So for variation, there should be the mixing of the genes, and that only happen when there is a cross pollination, right? Next question, what is self-incompatibility? Why does the self-pollination not lead to the seed formation in the self-incompatible species? This question is again coming from the section 1.2 of the NCERT. Let us see the answer. Self-incompatibility is the genetic mechanism in the angiosperm that prevent self-pollination, right? Self-incompatibility we have just now discussed about. This question is asked in the different framework, right? So they prevent the self-pollination. It develops genetic incompatibility between the individual of the same species or between the individual of the different species. Exactly same species from the same flower. If the pollen is coming, then they will discourage it. They will not allow the pollen to germinate on the stigma. Or if the pollen is coming from the different flower, then also it do not allow the pollen to germinate on the stigma. The plant which exhibit this phenomena have the ability to prevent the germination of the pollen grain and thus prevent the growth of the pollen tube on the stigma of the flower. So if they are not allowing the pollen grain to germinate on the stigma, then there is no question of development of the pollen tube. Pollen tube. If there is a germination, then only the pollen tube will be formed. And if there is no germination, then how could the pollen tube will be formed, right? This prevent the fusion of the gametes along with the development of the embryo. So there will be the no fusion of the gametes. Okay, because no pollen germination, no fusion of the gametes, and there will be no formation of the embryo as well. As a result, no seed formation takes place. So in this way, the self incompatibility is protecting to this inbreeding depression, and they do not allow the self pollination to occur. Next question, and this is the last question. What is bagging technique? Very, very important. And how is it useful in the plant breeding program, right? This question is also coming from the 1.2 section. This 1.2 section is very long in this particular chapter. Sexual reproduction is flowering plant. You can see this first from question number 1 to 10, all coming from the section 1.2 only, right? So this was the question, and let us see the answer. Various artificial hybridization technique under the various crop improvement programs involves the removal of anther from the bisexual flower without affecting the female reproductive part pistil through the process of emasculation. So if you are having a bisexual flower, the flower which is having male reproductive organ as well as the female reproductive organ, okay, both the reproductive organs are present in the same flower. So for the breeding technique, you want that some particular type of pollen grain should be brought on this particular flower which you have selected, bisexual flower. So what you will do, if you allow another pollen grain that you have selected one, the ones you have selected, you put it, then there could be the chances of that the pollen grain from the same flower could also come to the stigma. So how to stop this? First of all, you will select that particular flower which is having both that parts, male and female reproductive organ, and be ensure that the female reproductive organ is not at a maturation phase, it is not got matured, right? And at that point you will remove all the anthers from that flower. Now this flower is having only the female reproductive organ, this means that this is the female flower, right? So anther from the bisexual flower without affecting the female reproductive part pistil. Remove all that anther, and that removal of anther is called as emasculation. You will do with the help of forceps. With the help of forceps, one by one, you will remove all the anthers, and this process is called as emasculation. Then these emasculated flowers are wrapped in a bag to prevent the pollination by the unwanted pollen grain. Now this flower is having only female flower, right? Female part. Now you will cover it with the bag, and your bag should be made up of the butter paper, right? Butter paper. So why we are protecting this female flower with this butter paper bag? Because to prevent the pollination by the unwanted pollen grain. Unwanted pollen grain could come with the help of some biotic factors or the abiotic factors. So you will cover up with the bag, and this is called as bagging. This is called as bagging. So this process is called bagging. This technique is an important part of the plant breeding program as it ensures that the pollen grain of only desirable plants are used for the fertilization. You have some chosen plant which you want the pollen grain from that particular flower only brought and get pollinate that flower which you have made it a female flower, right? You have some desired plant variety, desired pollen grain, that with the help of brush you will put it on that particular flower which you have emasculated, right? And ensure that it should be rebagged again. It should be rebagged again. Okay, so this is all about today's session. We have discussed about 1 to 10 question from the NCERT book. Rest of the question we will discuss it tomorrow. So we will meet again tomorrow and discuss the rest of the question from the NCERT, right? So thank you very much for attending today's session. We will meet tomorrow.